biology lovers, welcome to Cell to System. Today we are going to discuss about the exciting series of phenomena that occur after the fertilization. So without wasting any time, let's begin. Zygote, this undergoes mitotic division to form embryo. This is the first change that will occur. This zygote, it will undergo mitotic division and form embryo. This embryo, it has mainly three parts. One is radicle, another one is plumule and cotyledons. It forms the root system. When the embryo will germinate to form a new plant, the root system will be formed by the radicle. The plumule will form into shoot system. These cotyledons, they will form the seed leaves. Leaves or the protective parts. This is the fate of zygote. Now what happens to the endosperm? This primary endosperm nucleus or PEN, it forms endosperm. Endosperms are of three different types. We will understand it when we will study the formation of endosperm. Now, this endosperm, it is responsible for the nourishment of developing embryo. Talking about the ovule forms seed. The integuments of the integuments form the seed coat. That is the outer covering or the outer dark covering of the seed is known as seed coat. Don't confuse seed coat with the cotyledon. We will differentiate all the things whenever we are going deeper into the discussion. So ovule, it changes into seed, and the integuments, it changes into the seed coat. What about ovary? The ovary enlarges to form fruit. The other structures, like some other structure like synergids or antipodal cells, they degenerate because now they don't have any function. Other floral parts like sepals or petals or stamen. In case of pistil, only the stigma and style, they all degenerate or fall off. This is the basic things, the basic changes externally we see in a flower after the fertilization. Now let's go into deeper and we will dive into the formation of embryo. We have come to the part that is formation of embryo. This formation of embryo occurs differently in case of monocots and dicots. To some stage it is same in both the plants, but after further division of the embryo, it has different structures in them. So let's understand. Now this first division, it occurs in both the plants, this zygote, it goes in asymmetric division, asymmetric mitotic division. Asymmetric because this basal cell is large and this one is terminal cell is small. This terminal cell, it is responsible for the formation of pro-embryo, and basal cell forms the suspensor. Suspensor pushes the pro-embryo towards the endosperm. So pro-embryo is the main structure which will further develop into the embryo, but this suspensor, this pushes the embryo, pro-embryo to the endosperm. So this time the zygote, it forms two cells. This is the basal cell, larger one, and this is the terminal cell. This is the terminal cell or this is the basal cell. It will finally grow into the pro-embryo and eventually pro-embryo will form the embryo, and this is the cell which will form the suspensor. So now let's understand what happens next. The second stage is pro-embryo stage. In this stage, terminal cell, it forms the pro-embryo and the basal cell forms the suspensor, as I said earlier. So let's not waste time in here. Now the next thing that is different in both dicot and monocots, that is the formation of embryo. Formation of or development of embryo in dicots. The development of embryo in angiosperms in dicots occurs mainly in 4 stages. We have first stage is globular stage. Here the pro-embryo or the terminal cell, it is undifferentiated. It is globular in shape and the suspensor is as usual present. This is the suspensor and this is the pro-embryo or the terminal cell. This is the globular part. This stage is known as globular stage. Next is heart-shaped. As we all know, in the heart-shaped stage, the appearance of heart shape is due to presence of or the formation of 2 cotyledons in case of dicots. So this part, this embryo, pro-embryo or terminal part, it forms the 2 cotyledons . And this is the suspensor. This is pro-embryo. Third one is torpedo stage. In this stage, the 2 cotyledons are visible and as well as the plumule and radical part is also visible. So this is the torpedo stage. This is the suspensor. These are the two cotyledons. Hope you can see or differentiate this is the radical and this one is plumule. Radical will finally form the root system and plumule will form the shoot system. Fourth or final stage or changes in the development of embryo in dicots is the mature embryo formation. After going through these three stages, the embryo will form into mature embryo. This mature embryo, it consists of cotyledons, which is 2 in number, embryo axis, in which the embryo will be situated. Embryo axis will consist radical and plumule. Another one is epicotyle and hypocotyle. These are the parts of the embryo or the mature embryo formation. Development of embryo in monocots. In case of monocots, the zygote is differentiated into terminal cell and a basal cell. In case of monocots, there is no such structures of globular stage, heart-shaped stage, no such structures found. But in case of them, the terminal cell, it is differentiated into two cells, top cell and middle cell. And this basal cell is responsible for the formation of suspensor. Here the suspensor is very short-lived. This is very short-lived. Now this top cell is differentiate into plumule and cotyledon, and the middle cell, it forms the radical and hypocotyle. This plumule, it forms again shoot system, and the cotyledon forms one cotyledon here. In case of monocots, it forms the root system and it is situated between the radical and the cotyledons. So this is all about the embryo development. Now let's start with the development of endosperm. The endosperm, that is the nourishing tissue of plants or the nourishing tissue present in the seed. This endosperm, it can be of three types, differing from one species to another species in the angiosperms. So these are Now we need to understand why these endosperms are of three types. Why this differentiation? The differentiation is because of their developmental pattern. First, starting with the cellular endosperm. In case of cellular endosperm, the primary endosperm nucleus or endosperm mother cell, it mitotically divides to form two daughter cells. These two daughter cells, among them, in one cell the further division occur and this look like that, and after further several divisions the cellular endosperm is formed. Now this is seen in, or this is found in, datura, magnolia, etc. This is the structure of cellular endosperm. Now starting with halobial endosperm. To halobial endosperm, the endosperm mother cell, it divides into two cells, one at the micropylar end and one at the chelazal end. Now in case of this micropylar end of the cell of the endosperm, free nuclear division occurs, as you can see, but here no free nuclear division occurs, only it divides to form two cells. Here again, after free nuclear division, it forms cell wall around the nucleus at the micropylar end, but at the chelazal end there is only 2 to 3 cells are formed. No further cell wall formation occurs. So this kind of structure of endosperm is known as halobial endosperm, and it is found in water lily or nymphia. Water lily, which has a scientific name nymphia. Now the final one, nuclear endosperm. Here in nuclear endosperm, the endosperm mother cell, it undergoes open nuclear division and it will form a central vacuole. After that it will form a central vacuole. After the formation of central vacuole, the nucleus, each nucleus will form a cell wall. So this is the structure of nuclear endosperm. It is seen in maize, sunflower, coconut, capsula, etc. So these are some examples of nuclear endosperm. Now this is all about the structure of endosperm, and in today's lecture we have studied or we have discussed about the main post-fertilization changes, elaborating the formation of embryo and endosperm. In the next class we will be knowing about the formation pattern of seed and fruit. Now this is all about today's discussion, and if you really like the video, please subscribe to Cell to System, and to continuously study with me, please press the bell icon. Thank you.